Okay, I'm Barry Lester, and I have it on my notes to say that the title of this workshop is Maternal Lifestyle Study, MLS Overview, Measures, Results, and More, Session 1. This is the outline that I'm going to use for this presentation. I'm going to talk about the historical context in which MLS was developed, the study design, summary of the overall findings, and then I want to go through the different types of findings because we started out looking at, for example, main effects, and as the study went on, it branched out into looking at other ways of studying prenatal cocaine exposure. And then I'm going to end with some conceptual thoughts about how to maybe think about MLS in the current scientific zeitgeist. So first, let's look at the historical context. These were headlines from 37 years ago. Some of you may have not been born. Some of you were probably kids, teenagers. But you can see that the early reports in the 1980s really created a huge public frenzy about unfit to parents women and how their babies were going to be damaged, and the nickname crack babies came up. And it was obviously considered a huge epidemic. And again, if you just look at these headlines, mothers turned into monsters, the future looks bleak, etc. And so, basically what happened was there was a lot of insufficient and inaccurate information. Society really overreacted. Women were prosecuted and children were removed from birth mothers. Families were split up. It had a huge impact on the whole social structure of the country. And part of the problem was that a lot of the work was based on animal studies where the dosing of cocaine that they used was nothing like you would find in the human condition. I mean, the doses were hundreds of times larger. And that's where the initial fears came from. And then, people started noticing things in NICUs, and there were a number of single case studies reported. I remember one very clearly. There was a case study of a baby who was cocaine exposed and died of sudden infant death syndrome, followed by a headline, cocaine causes SIDS. So by, there was a tremendous amount of anger and hostility at these pregnant women. And, again, going back in history and in context, this was way before the whole idea that addiction was a disease of the brain rather than about, malicious, uncaring mothers hurting their babies. I mean, that concept didn't even exist then. But, by the mid-1990s, the number of children in foster care reached an all-time high of over 500,000, and many of these children suffered emotional problems. They would be moved from one foster home to another. Their attachment and other relationships would be compromised, and they would develop emotional problems, and then people would say, see, this is what cocaine does. Well, I'm sure I don't have to tell you what that kind of changes in placement can do. A number of studies were launched by the NIH, probably 20 or so. MLS was the largest, and results started coming in. And what we found out was that the effects were not nearly as severe as what were expected. Linda Mays, coined the phrase a rush to judgment. And again, now you saw the pendulum swinging in the opposite direction. Crack kids not broken. Mother's cocaine use may not doom the child. And the blue headline is our initial MLS findings. Again, because MLS was the largest of all the prenatal exposure studies, that major trial finds only subtle fetal injury effects from cocaine. That turned out to be a really important, I don't want to say buzzword, but the term subtle became used a lot in different kinds of contexts, and it was misunderstood a lot. And that kind of prompted this article trying to explain the meaning of subtle effects. So we did a meta-analysis of studies of IQ and receptive language and expressive language. This was in school-age children. You can see there weren't a lot of studies at the time. The mean IQ difference between exposed and unexposed was 3.26 IQ points. That's a small effect size. The language studies showed a larger effect sizes. But what we then did was we translated these increases in the number of children that would fall into either 2 or 1.5 standard deviations below the norm. We calculated the additional increase in number of kids to society, and then the cost of society, an additional cost, because most of these kids are going to wind up in early intervention. So, what these published follow-up studies showed was that yes, cocaine is associated with, reliable decrements and cognitive performance, and they're subtle in 2 ways. If they're subtle in that they are small magnitude, especially if you look at the IQ findings, and the small magnitude is in sharp contrast, obviously, to what was being reported in the media. The effects are also subtle in the sense that you find larger effects in more subtle domains, such as language, than in more global measures, such as IQ. But the public health consequences of these effects are substantial, so that the real take-home message here is that we should not equate small magnitude with lack of importance. So even though cocaine exposure may not cause this devastating brain damage, it certainly can result in changes that are going to increase the need for special education and at a substantial cost of society. It's also been said that if you took that money and used it for prevention, you could probably eliminate a lot of these deficits in the first place, or at least reduce the order of magnitude. But prevention is always difficult to sell. So the MLS study design itself, MLS was originally designed specifically to look at the effects of prenatal cocaine exposure, that is, main effects. And then we went on to look at other ways of thinking about prenatal drug exposure, as we'll see. So it was a longitudinal study. It began in the early 1990s, about, amid the climate we just went over. The children were followed through age 16, and funding was complicated. The initial funding was through a cooperative agreement, a U-10, through NIDA, NICHD. There was additional support from ACYF and CSAT. And then there was a separate neurodevelopmental contract from NIDA to develop the neurodevelopmental measures and battery. And then later, the contract was over, and so the study was funded by a series of R01s from NIDA, and there was some additional funding from the NIMH. This is the organizational structure of MLS. It was conducted in 4 sites: Miami, Wayne State, University of Tennessee at Memphis, and Brown. Brown was also awarded the Neurodevelopmental Data Center, or the battery. And initially, as you'll hear, there were 2 phases, phase 1 and the follow-up phase. The NICHD NICU Research Network was the original data center, and then it was later taken over by RTI, and you'll hear from Carla from RTI tomorrow. So as I said, there were 2 phases of MLS. There was a newborn phase from which we collected medical data and identified subjects. So we screened 19,000. There were almost 17,000 eligible, almost 12,000 consented, and of those, 10% or 1,185 were exposed. So these were the candidates for enrollment in the longitudinal study, and I'm going to be very brief about discussing these findings because we really want to emphasize this webinar on the longitudinal follow-up. So we're going to focus on the longitudinal study, which began at 1 month with 1,388 subjects. Now, we knew it then, we know it now. Most women who use cocaine also use other substances, alcohol, marijuana, and tobacco, and that the real problem, and what we later looked at, is polydrug abuse. But the idea was, if we looked at a group who were cocaine exposed and exposed to other substances versus a group that were not exposed to cocaine but were exposed to other substances, the differences between the 2 groups, obviously with covariates, could be attributed to cocaine. So we have, of the 1388, there were 658 exposed and 730 not exposed to cocaine. The groups were matched on prematurity, race, and sex. The cocaine-exposed group, again, had cocaine and or opiate use during pregnancy, and that was determined by self-report and or meconium toxicology. We'll get to that in a second. And the unexposed group had no cocaine or opiates during pregnancy, but they were exposed to what we call background substances: alcohol, marijuana, tobacco. We followed them through age 16, and we had 75% retention at age 16. The demographics of the follow-up cohort was representative of the population. They were mostly Black. Cocaine-using mothers are often older. That's fairly typical. Unmarried, on Medicaid, less education, and the gestational age was similar because that was a matching criteria. So overall MLS summary findings, let's get into. Before MLS even started, prior to phase 1, we developed the methods to analyze drugs in meconium. And that's important because the assay included the confirmation, GCMS confirmation, following a positive screen. problem with screens is that they have a high false positive rate, and that rate is higher for some drugs than others. But what that does is it leads to over-identification of exposure. So studies that do not include a confirmation, which was true of many of the early studies, have a less reliable detection of prenatal exposures. And again, this was shown in this study from MLS phase 1 here. This is our report, our initial report, where we found that your identification of exposure is improved. It's much higher, more accurate with GCMS confirmation of meconium, and also when it's coupled with a positive endorsement from the mother. So very brief summary of phase 1 findings, and again, this is what was in that headline. The kinds of medical problems that were reported, thought to be, were rare, less than 5%. Many of them were not related to cocaine. And similarly, the physical damage of the infant was rare and not related to cocaine. So that's examples of that would be cardiac deficits, brain injury. The cocaine-exposed babies were born only 1.2 weeks lower gestational age and 536 grams lower in birth weight. So none of that is terribly dramatic, and again, unexpected. So if we look at the MLS publications, there are 88. You should have gotten a list of them, and of the 88, 44 tested the main objective, or the specific aims of MLS, which were to determine the effects of prenatal exposure on child outcome, controlling for other prenatal exposures, medical, caregiving, environmental factors. This shows the various domains that we looked at in MLS, and the domains by age groups, birth to 3, 14 to 11, and 12 to 16. This is just a little plug for MLS that I'll mention more than once. This is a very rich data set. I mean, it's unique in that some of the domains here are covered in virtually all of the longitudinal studies, but you're not going to find all these domains covered in a single cohort other than MLS, and virtually at each age. So this is a unique opportunity to look across domains. Covariates, of course, are critical, and we used a number of covariates depending on the hypothesis of the study, but these are probably the ones that were most commonly used, and typically, we would control for these factors and then look and see whether or not they also had their own level of statistical significance. So of the 44 studies that did look at main effects, 82% of them did show main effects, and they showed main effects of cocaine exposure, prenatal exposure, really across a wide range of domains. If you look on here, many different kinds of domains. So the effects were not confined to any one particular area or any few areas. They're fairly broad-based. 8 of the studies, 18%, did not show main effects. And again, they're typically across different areas, and once again, not specific to any one domain. So let's look at some examples of these main effect findings, some specific studies. When MLS was starting, I mentioned that we had this neurodevelopmental data center, neurodevelopmental battery. So we developed this exam, the NICU Network Behavioral Scale, to provide a comprehensive assessment of neurobehavior in at-risk babies in general, but especially those who were drug exposed. And the exam can be used from birth to 1 month of age, longer if it's a preemie. The battery also included the acoustical analysis of the baby's cry. We recorded the cry during the administration of the ends, and then looked at the acoustic characteristics, and that was really initial findings where we showed that oops, okay, I'm very sorry. What did I do? I should know better than to touch these things. Well, I seem to have lost I think I'm going to need some help here.
Well, maybe while you're figuring things out there, Barry, if you can hear me, I can answer one of the questions that we have here.
Yeah, you can try. I'm not sure I'm going to be able. Oh, wait a minute.
No, I said I could answer. Oh, are you back to where you want to be?
I think I am.
Okay, great.
How's that? Okay. So our initial findings, we found cocaine-exposed infants had lower scores on some of the end scales, and also differences on the acoustic cry analysis. We then looked at ABR, auditory brain response, and we also found that neurotransmission, some of the interpeak latencies were affected by prenatal cocaine exposure. Now, for some reason, I can't. Okay. I'm very sorry, folks. Okay. So the ends itself has 12 summary scales, and what we did was, and this was later work, but it has these 12 summary scores, and what we did was we used latent profile analysis and converted the summary scores into these mutually exclusive profiles. 6% of these were in this particular profile, and this profile is really abnormal behavior, and I won't go through it very, very detailed, but basically these babies have a hard time tracking. They need a tremendous amount of handling, very poor self-regulation, highly aroused, highly excitable. They're hypertonic, very poor movement, and a lot of stress. So this profile 5 here was related, was found mostly in the cocaine exposure group. And then we looked at the percent of infants with abnormal development. So those who had this abnormal profile, the same profile you just saw. And we're looking at the proportion of infants with poor developmental outcomes starting at 1 year and going through age 4.5 on IQ. And so you can see the highest proportion of babies with abnormal outcomes or adverse outcomes come from the same abnormal ends profile. Here's another example of a main effect study. So here, this is at age 7. The exposed kids were more likely to receive a referral for special education, both at ages 7 and 11. This is looking at externalizing problems on the child behavior checklist, CBCL. We had done some other work where we looked at the effects of cocaine exposure, particularly on externalizing behavior at 3. And here we're going from 7 to 13 using both parent report and teacher report. So the parents reported the most behavior problems, externalizing problems in the babies with high levels of prenatal cocaine exposure. We did a couple of neuroimaging studies. This is fMRI, functional imaging, and I don't know if you remember Pokemon. So we use Pokemon for a no-go, no-go task. And the child is instructed to be sitting in front of a computer and told to push a button every time they see a Pokemon character, except Meowth. Meowth was the bad guy. So if you see Meowth, do not hit the button. So clearly what we're looking at here is response inhibition. And we're looking at the right caudate, left occipital gyrus, and right frontal gyrus. The orange or red here shows the areas where the cocaine group has more activation than the unexposed group. And the blue is where the unexposed has more activation. These regions are important because they're related to controlled attention, response inhibition, and essentially what we thought was going on here was that the reason the cocaine exposed kids have higher levels of activation is because it takes more activation, brain power, if you will, for the exposed kids to accomplish the same tasks as the unexposed kids. Here's the other imaging study. This is looking at MRI structure, and what we found here, we're looking at the right dorsal prefrontal cortex and the right ventral medial prefrontal cortex over here. And what we find is that the right prefrontal cortex is thinner in the cocaine exposed. So what's kind of interesting about these two studies is that they're showing that both structural and functional changes are associated with prenatal cocaine exposure. Next, we're going to look at interaction effects or mediation effects. So these are studies in which there are interactions between cocaine and other variables, and studies where the cocaine effects are mediated by other factors using techniques such as structural equation modeling. So here we looked at saliva before and after we collected saliva before and after a stress procedure. These kids are now 11, yeah, and we used the Trier to elicit stress. We divided the kids into those who showed an increase in cortisol and from baseline to post-stress, and those who showed a decrease or blunted cortisol response, reactivity to stress. And the cortisol reactivity to stress was more likely to be blunted in children with prenatal cocaine exposure. So that's this group and this group. But you can see the interaction with domestic violence, because the exposed kids exposed to domestic violence showed the strongest effect. You can see the differences here versus here. Now, we'll get into this a little bit later, but the cortisol blunting is important and interesting because it is thought to be due to chronic long-term stress, what Bruce McEwen calls allostatic load, which is the wear and tear of the body produced by the repeated activation of the HPA axis and related biological stress systems. So you're basically beating on the neuroendocrine system over a long period of time until it gets worn down. And so the cohort response, the HPA response, is unable to respond in the way that it should. Here's an example where we're starting to look at SEMs, and we're looking at relations between prenatal cocaine exposure and arrests at 15 years of age. So initially you have what is here a direct effect, statistically significant effect. However, when you introduce a measure of inhibitory control problems, what you find is that prenatal exposure is related to inhibitory control problems, which in turn is related to arrests at age 15. The cocaine effect is still here, but it's diminished. It goes from 7.4 to 6.7. So this is an example of partial mediation where the effect of cocaine on arrest is partially mediated by inhibitory control problems. Now, inhibitory control is it's really part of a larger construct called neurobehavioral disinhibition, and much of this is based on the work of Ralph Tarter. And it refers to an underlying deficit in behavior control, but it's really thought of or represented as a single dimension or a factor. And it has a lot of different kinds of problems on it. I mean, these things hang together. They don't occur in isolation. So impulsivity, reactive aggression, sensation seeking, but also impaired executive function. So sometimes you get a single neurobehavioral dimension. Sometimes you get one that's more behavioral, with a second one that's related to cognition, particularly executive function. And this inhibition is related to substance use in adolescence, early age onset of substance use disorder, and substance use in adulthood. So we took some of our measures. These were the actual measures that we used, and we constructed a neurodevelopmental factor score. So we had an ND factor, and what we were hypothesizing is if we look at this disinhibition factor, and we computed this at ages 8 to 9, 11, and 13 to 14, that prenatal cocaine exposure would result in or be related to neurobehavioral disinhibition, and that would in turn be related to substance use onset with, of course, covariate. So we're hypothesizing a mediation here. So we measured substance use every year from ages 8 to 16. These are Cox regression survival curves, so that the y-axis shows the percent who did not use. When you look at the unadjusted analysis up here, the hazard ratio indicates that children in the exposed group are 42% more likely to use drugs earlier than children in the unexposed group. That's the yellow versus the red. When you then add in the disinhibition score or factor, when you add it to the model, the effect of cocaine is no longer significant. But the higher disinhibition factor is now related to earlier use. So that's again suggesting that the role of disinhibition plays the role of a mediator and essentially replaces or knocks out the cocaine effect. It also turns out that child abuse as a covariate, those kids with a history of child abuse were more likely to use, in this case, tobacco at an earlier age. We looked at also age of first marijuana use, and it's the same story. I don't necessarily have to go through the same thing for you, but again, unadjusted, you get a cocaine effect. Adjusted, you get the disinhibition effect. Now we get a covariate effect for boys and for community violence. Similarly, if, we looked at illicit drugs, that includes marijuana inhalants, cocaine, et cetera. And so if you look at any illicit drug use, the story is again, pretty much the same. So let's move on now to polydrug effects. And I mentioned earlier that even though we were looking at main effects of cocaine, we knew that the real problem, quote unquote, real problem, was polydrug use. And so we moved to conduct a series of studies to look at exposure from a polydrug use perspective. In the first of these, we had found earlier differences we saw before on a number of measures, including the ends. So here, this is using structural equation modeling. We found that we now have a composite measure of prenatal drug exposure. So we're not just looking at cocaine, where we have this composite factor. And this composite factor was related to dysregulation on the ends and to problems in arousal and stress abstinence, which continued. So the behavioral effects were then related to temperament on the infant behavior questionnaire, particularly distress to novelty and distress to limitations. And these were in turn related to problems on the CBCL at age 3 and age 7 based on both teacher and parent report. The other thing that's interesting about this is that there are, what I just showed you are a series of indirect effects, but there are also direct effects. You can see prenatal exposure is related to both 3 and 7 year CBCL independent of this other pathway. There's also a pathway here through SES. So in a sense, what we think this is saying is, there's some things that are, let's call them fixed, but there are others where you can see opportunities for intervention. You can say, oh, here's a place I can intervene, here's a place I can intervene, and potentially reduce the magnitude of behavior problems. Here we were using multivariate growth modeling, and we're looking at relations between prenatal exposure and early adversity. So this is based on just a cumulative sum of the number of drugs that you were exposed to. And in this case, we split the neurodevelopmental factor into both a behavioral factor and an executive function factor. As I mentioned before, a lot of the literature includes both factors. And the prenatal exposure and early adversity predicted the trajectory of behavioral dysregulation from 8 to 9 to 13 to 14. The effects of prenatal exposure and early adversity on executive function, which you see here, were mediated by behavior dysregulation at 8 and 9. So what this is telling us is that some of the variance in behavioral dysregulation is probably an important precursor that influences these changes in executive functioning difficulties. And, you can think of this in the context of the later maturation of the prefrontal cortex areas, and we did see brain-related effects early on that are associated with executive function, and that, it suggests that there may be a different rate of development for the neurobehavioral disinhibition and executive function systems. Here we have initiation of alcohol use by age 16. And we're finding, you can see, if this is the same summary of the number of drugs the infant was exposed to, and we see that that's related to early adversity, which is in turn related to executive function and initiation of alcohol use. So there are two other things that are noteworthy here. One is that the exposure plus adversity combination goes along with the idea of prenatal plus postnatal stress, a model that we will discuss later, thinking about prenatal exposure as a stressor. Second thing is that now we have cortisol and respiratory sinus arrhythmia, aka vagal tone, also playing a role affecting the executive function and then initiation of alcohol use. So we now have the addition of physiological measures and another example of the richness and diversity of the MLS database that would even enable us to look at something like this. As MLS went along, we became interested in resilience and protective factors. We were asked about that a lot. Why don't you look at good outcome for a change? So we did, and in one of the studies where we're looking at 11-year-olds, we looked at two protective factors, relatedness and effortful control. Relatedness to others is measured by various indicators of positive relationships and pro-social behavior to teachers, parents, friends, and peers. And effort for control was measured by inhibitory control and attentional functioning measures. And so what you can see here is that both positive relatedness and effort for control reduce the incidence of conduct disorder and ODD. Relatedness reduce the incidence of school delinquency and depression, and effortful control reduce the incidence of crimes against people, general delinquency, and ADHD. At age 15, we looked at trajectories of behavior problems from ages 5 through ages 15. Again, you saw something similar before. And again, the fact that you can do that, the fact that we have, repeated measures of these kinds of behavior dimensions, 5, 7, 9, 11, 13, and 15 is another unique aspect of MLS. We looked at 4 categories of prenatal drug exposure, and the categories were high prenatal cocaine exposure with other drugs, prenatal cocaine exposure with other drugs, no prenatal exposure with other drugs, and neither prenatal exposure nor other drugs. And then we looked at risk and protective factors. We looked at them at the individual, family, and community levels. And, not surprisingly, high levels of protective factors were associated with declining trajectories of problem behavior scores. So that, that's a good example of a protective factor. I mean, you can see here that if you're high risk, low protect, you have the highest number of behavior problems, likewise over here. But if you're in the low risk, high protect group, you can see that your trajectory for behavior problems decreases over time, here, 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 and here. So, some conceptual thoughts. We started out this presentation by looking at MLS and the context in which it started. So I'd like to conclude by thinking about MLS in the context of some of the current scientific thinking, hoping that this might help you formulating questions on your own that lend themselves to using the MLS database. Because we do think about the world a little differently today than we did in the 80s and 90s. Some of it is similar, some of it is different. But let's see. So typically when we think about the effects of cocaine, we think about two kinds of effects. One is neurochemical, where cocaine blocks the reuptake of dopamine and increasing circulating levels of dopamine. And the other is vasoconstrictive, where cocaine increases pressure on intrauterine arteries, reduces cerebral blood flow, reduces nutrients, and that's a vasoconstrictive effect. So these are different and have been studied in a number of ways. But we might think about cocaine from a different perspective, and if it works for cocaine, it's going to probably work for other drugs as well. And we think of this as a third pathophysiology of cocaine, that in addition to the neurochemical and vasoconstrictive, that there may be a third pathophysiology in which we view cocaine as a prenatal stressor. It's a stress. Drugs are stressful in the intrauterine environment. And the consequences of that are to alter fetal programming, which we'll get into in a moment. And what that theoretically would do is alter gene expression. It'll alter the expression of key placental genes, particularly those that are involved in the stress axis, the HPA axis, and disrupt the HPA axis and result in neurobehavioral dysregulation in the newly born exposed infant. So where does this come from? This whole idea, in part, comes from what became known as the Barker hypothesis, or fetal origins, now called developmental origins. And during World War II, the Nazis starved the Dutch during the winter of 1944 and 1945. And what happened was if you follow these kids until they're full-grown adults, you see that there was a relationship between birth weight here and the prevalence of diabetes and glucose tolerance in men. And in the case of women, birth weight was related to obesity and an increase in metabolic syndrome. Oh, how'd that happen? Why'd that happen? So the idea behind the fetal origins is that susceptibility to cardiovascular disease, diabetes, other kinds of problems is programmed in utero as a response to fetal undernutrition. So what do we mean by that? Well, what we mean by that is that one of the things we know about our species is that because of developmental plasticity, the organism can change or reprogram, can actually reprogram structure and function in response to environmental cues. And those cues can be internal, intrauterine, as well as postnatal, which will bring us to environmental adversity. And that's very adaptive because it means that you can have a whole range of phenotypes developing from a single genotype depending on environmental factors, again, pre or postnatal. So basically what happens is the fetus receives signals from the intrauterine environment, and that stimulates a recalibration of fetal physiology. And that results in a phenotype that's adapted to the postnatal environment. what happened in the case of the Dutch famine is that these babies were undernourished, right? They were starving. So the insult here was undernutrition. And so the signals about undernutrition were read by the fetus, and metabolic systems, metabolic pathways were reprogrammed because the assumption was that if I'm in a malnourished environment, then it would be much more beneficial, not that fetuses talk like this, but it would be much more beneficial to be prepared for that postnatal undernourished environment. Well, what happened was the famine ended, and in fact these babies were born into adequate environments, normal nutrition, but their metabolic systems had already been reprogrammed, and so they essentially couldn't handle there were that they were expecting not to have a lot of food around, and there was a lot of food around, so they weren't prepared and they gained weight rapidly, and that's the rest of the story. So what that raised is, well, if undernutrition can do this, what about other kinds of insults, like prenatal cocaine or other drug exposure? What mechanism would do this? And the idea was advanced that it could have to do with epigenetics, and that epigenetics could provide the actual molecular basis for fetal programming. So I'm not going to go into detail about epigenetics because I'm going to be pulled off the stage. But epigenetics refers to changes in gene activity or gene expression without affecting the basic structure, the genetic code. So the gene stays the same. The gene doesn't change, but it doesn't do necessarily what it was programmed to do. And genes can be silenced. You've heard of switching genes on and off completely during fetal development. That's epigenetics. It's the mechanism is methylation, DNA methylation, which directly affects gene expression, so that when genes are completely methylated, they are literally shut down. But it's not a light switch. It's more they can be calibrated. You can increase or decrease gene expression. So people use the phrase that genes load the gun, epigenetics pulls the trigger. So this is a model that Jim Padbury and I developed as a way to think about fetal programming and stress and epigenetics. The idea being that if we think about drugs now as an intrauterine stress, adverse maternal environment, again, it could be drugs, toxicants, smoking, other stressors, and that these stressors are going to affect the activation and the circulating levels of glucocorticoids, which we will know as cortisol. And there are two primary genes that we look at. There are others, but there are two primary genes. One is the glucocorticoid receptor, which affects the number of sites that cortisol can bind to, the number of receptor sites. So if this gene is methylated, there are fewer receptor sites for cortisol to bond to, and you get an increase in circulating cortisol. 11 beta is a placental gene that converts cortisol to its inactive innate form called cortisone. So if this drug is methylated, it will not convert as much cortisol to cortisone, and again, you'll get more circulating cortisol. So these two genes work in reciprocity to regulate the amount of cortisol that the fetus is exposed to. And when something goes into disarray, such as an insult like cocaine or any other stressor, it will increase methylation of these genes and increase levels of circulating cortisol that dysregulates the HPA axis and newborn neurobehavior that we see on the ends. Is there any evidence for this? Yes. There are 17 published studies looking at epigenetic processes in placental genes and human neurobehavior, primarily on the ends, and those studies are reviewed here. So what that brings us to is an overall way of thinking of MLS from the point of view of a stress model, in which we would say that prenatal cocaine exposure and environmental adversity are both going to, through epigenetic mechanisms, they're going to affect the HPA axis, resulting in a predisposition to neurobehavioral disinhibition and affect developmental outcome. And then we would invoke other stressors, fetal origins, and also the notion of toxic stress. I mentioned Bruce McEwen before, and so the idea that frequent and prolonged activity banging away at the HPA axis results in wear and tear, and it reduces the ability of the HPA system to react. We're also saying here that there may also be independent effects of prenatal exposure on outcome, and these could be neurochemical and vasoconstrictive, and there may be environmental effects on developmental outcome, for sure they are, that are not related to cocaine. So, last slide, next to the last slide. So we would argue that these that developmental influences during the prenatal period, they're associated with adverse outcomes. Yep, they do not occur in isolation, and that's where we get our neurobehavioral disinhibition construct, our disinhibitory behavior, that prenatal substance exposure, cocaine or other drugs, can affect the fetal programming of these stress-related pathways, epigenetic changes in the stress-related genes, dysregulation of the HPA system, newborn neurobehavioral organization, which really gives you an increased susceptibility, is another current word in use, to postnatal environmental adversity and the development of later behavior problems. And, you know, I very much like the work of Jack Schoenkoff, Tom Boyce, and Bruce McEwen, who talk about the way, and again, this is another, let's say, popular construct, that early experience can affect adult health in two ways: by the biological embedding of insults during these sensitive periods, and by accumulating damage over time to chronic stress, aka allostatic load. And they put this in the context of the fact that substance use itself is a major risk factor that's included under the description of toxic stress, so it's considered a toxic stress, that was developed by the National Scientific Council on the Developing Child and is all part of this emerging idea of looking at chronic activation of the HPA and related stress response systems that result in these kinds of disorders. So special thanks to the NIH people who were so supportive of NIH, of MLS, and the principal investigators, myself, Henrietta Bada, Charles Bauer, and Seetha Shankaran. Tony Whitaker later joined us as a site principal investigator, and then the data center principal investigators, Linda Lagasse at Brown and Jane Hammond at the Research Triangle Institute. So that's it.
Okay, thanks, Barry, for all that wonderful information. We just have 10 more minutes of presentation that I am going to give about how to access the MLS data on the National Addiction and HIV Data Archive Program website. So hopefully Barry has really inspired you about what's available, and now you really need to find what it is that you need. So I've noticed that some of you have been asking questions, and you can feel free to continue adding questions, and we'll answer most of those towards the end of the presentation. I noticed that Sarah had a question about how many assessment time points there were, and hopefully my presentation will help address that. So let's see. First, I'm going to take you to the NADAP website and show you a little bit of what we have available there. When you visit the NADAP homepage, you'll see at the top of the homepage there's a search bar which you can use to search for data, and there's also a tab menu across the top for easy navigation. Also, something that this group might find interesting are the quick links for data users. They're towards the bottom of the middle of the home page. So to get to the MLS study home page, the easiest thing to do is just type MLS in the search bar, and that will bring you to a list of search results. We can click here on the Maternal Lifestyle Study. So now that you're at the study homepage, I'll just highlight a few areas that you might find interesting. We have the quick downloads link here, which can allow you to download all the study documentation, although I wouldn't necessarily recommend it because there's hundreds of files there. So you probably find it easier to navigate through the various data sets and select which ones you need. The table of contents here on the left of the screen can help you easily get to different parts of the study homepage. And I was going to highlight a little bit about the methodology. So first we'll just click here on methodology, and that will take us down to that portion of the website. And you'll see that we have details here about the sampling for the study, the modes of data collection that were used, and the extensive processing conducted by ICPSR. I want to highlight for you some of the study documentation that you might find helpful for finding information. So if you go up to the data sets portion of the web page, you'll see that there's DS0, and this means it's a study level file. So this is a comprehensive documentation file that has information on all the data parts. Let's see, download this. And let's see, I lost my navigation pane here. But let's see. Oh, I should have downloaded it earlier. It showed up differently on my computer. But I'll just go back to my presentation here because I've got the links there. So when you go into that documentation file, you'll see that there's a tab for the data set indexes, and you'll find this to be helpful. Unfortunately, it's not super clear in this slide, but what you have here is a list of all the assessments that were administered, and across the top, it's all the different ages that those assessments were taken. So it's basically a crosswalk of when those assessments were administered, so that can be a helpful tool for figuring out which data sets you'll be wanting to use. So there's also a source key that helps you identify which different group that assessment was given to. So there's parents, teachers, children, for example, some administrative files. Also in that documentation file, you'll find the table of data merges. So NADAP, because of the volume of data files that were available, the NADAP staff performed merges to make the curation of the data a little bit easier and also hopefully help you with the data use. So these files are grouped based on the source key codes that you can see in this column here. And if you look in the leftmost column, you can see the data set number, and we've tried to combine within the same phase and age data files with the same source key. So for example, you can see in data set 5, we have these two multi-source files that were merged together, and you can see the variable count for those files and the number of cases across all of them and by the individual sites for each data file. Also, you'll see that these are listed by the phase, and as you continue on through the other merged files, the age that was administered. Okay. So now if you, let's see if I can go back to the website quickly because I think that would be more useful. Okay, so now back on the Maternal Lifestyle homepage, if you go to the variables link here in the table of contents, that takes us down to the variables search feature. And so this will help you to search all the over 45,000 variables that are in all of the MLS data sets. So just to provide you with a quick example, I thought we could imagine that we're interested in searching variables about whether children were active in their communities. So to do that, I'm just going to type community into the search bar here. And that pops up with a list of results that shows there's 44 variables that contain a reference to the word community. Just to look at the first one here, we get a result of the variable name, the label, which says, is name involved in community? And then it will tell you which data set this was taken from. So this was a Maternal Lifestyle Study data set, phase 4, year 9, mother caregiver report data. So I previously checked the questionnaire for this variable to confirm that this was referring to the child, and the question states, is study child's name involved in any activities in your neighborhood or community? And examples of these are music lessons, sports, Boys and Girl Scouts camps, etc. So this does look like it's what we are looking for. So now this particular variable is available in multiple waves of the study. So we have this compare tool here that you can use to look across some of those waves. So I'm just going to select some here to give you an example. So we have also in year 10 the same variable was applied, and down here this one applies to year 11. So now that we've selected the variables we want to look at, we press compare. And this gives us detailed information about each variable with the frequencies. And so just for fun and a quick look, we can see that on the left here we've got year 11, then year 10, year 9. So the older ages are here to the left, and we see that the percentage is a little bit higher of people who said yes, they participated in a community activity for the older children than the younger children. So that's just a really neat tool to kind of just get an idea of what's available in the data set. So we have another search feature that I wanted to share with you, and that is for the related publications. So I also want to mention here that Barry said he has a list of associated publications that he'll be sharing with all the attendees, and I think some of those are new publications in addition to what we have available on our website right now, but those will be updated. So also, if you end up using the data for your own research, we ask that you please contact us and let us know about your publication. And you can do this by contacting ICPSR at bibliography@icpsr.umich.edu. And I believe I have that email address here in my slides. So that link here, these slides will also be available to you too. So we appreciate that if you let us know so that we can keep our records up to date and make sure that you get acknowledged for the work that you did. So as you might be aware, the Maternal Lifestyle Study is a restricted use study. So this means that it can contain indirect identifiers and presents potential disclosure risk. So our documentation files are available, but the data itself you'll need to apply to get access to, and you'll need IRB approval or exemption for that, and that requires that you complete a restricted data user agreement. So I'm going to give you a little bit more details about that process now. So applying for access to restricted use data, the first step is that you need to thoroughly review the documentation on the MLS website and really be careful to figure out which data sets that you want, because we just want to only be giving you the data that you'll need, because there's a lot of it. It's not like it could overwhelm you with how much there is, and also we just want to protect the respondents and make sure it's being used appropriately. So the more detail you can provide about that when you apply, the better off you'll be, and the quicker you'll be able to gain access to the data. So you want to make sure to also read the restriction notes and the access notes in the study description. And then once you're ready to apply, you know what you need, you can apply online using the access notes link in the study description. So to give you an idea about what the restricted data user agreement includes, you'll see that it's available as a PDF through the request system. It defines the requirements of the investigator and the investigator's institution, as well as ICPSR's obligations in providing the data. The RDU requires signatures from the requesting investigator, a legal representative of the researcher's institution, and this is somebody that can sign a binding legal document for the institution. And at an academic institution, this is typically a dean or someone higher. The RDU includes the information entered into our access system, the IRB letter of approval or exemption for the project, and a data security plan that is created through the investigator's responses in the request system. So that's something that you're going to be needing to think about. Like, once you have the data, how are you going to protect the data? So if you want to get a better idea of what that all means, you can see here at the bottom of the slide, there's a link for the restricted data user agreement example that you can take a look at, and this is also available on our website. So we have a lot of training available on our website to help you along in this process when you're actually ready to apply. You can visit the training tab to find a webinar about how to find and apply for restricted use data from NADAP, and it's easier than you may think. You can also visit the Find Data tab, and on that tab you'll see there's a link to the Access to Restricted Use Data webpage, and on that webpage we have both a PDF tutorial and a video tutorial about finding and applying for restricted use data. Oops, I didn't want to do that. On to the last slide here. So thank you to everybody who stuck it out here to the end. If you have any questions, you're welcome to ask them now. And we also welcome your questions at nadap@icpsr.umich.edu. If you have something that comes up after the presentation or you need assistance in applying for the data. Trying to see here if we have any new questions here. So Sarah had asked how many assessment time points there were. And while I don't know off the top of my head, I know that documentation file that I was referring to you on the study homepage. If you go to that data indexes table, you can see the complete list. Barry, do you know off the top of your head how many time points there are?
Yeah. Well, again, this is for the excuse me, follow-up phase. So 1, 4, 8, 12, 18, 24, and then every year after that through 16.
Okay. So we also have a question here about, were there any infant feeding outcome variables collected, e.g. breastfeeding?
We did, let's see, there was an early feeding publication. Lynn Legassi was the first author, so that'll be in your list of publications. I don't remember off the top of my head if breastfeeding was in there. I'm sure we collected it, nor do I know if it's in the public database. I mean, that information is available, right? I mean, you could do a search for breastfeeding, I would think.
Yeah, we can do a search for breastfeeding on the website, and I'd be happy to help that person with that if they need help with that. I can probably send you an email. Okay, let me see if we have any more questions here. I think that's, looks like it's about it. I'll hang on here for another minute or two just in case somebody has something that they didn't get a chance to type in yet. But other than that, we just really appreciate you all attending today, and that there'll be another MLS presentation webinar tomorrow, which I believe you can still sign up for if you want to find out more about the study. Is there anything you want to add, Barry, before we finish?
Yeah, I was just going to look up that feeding publication because I have them all. I have them all here. So let's see. Well, I guess I'm wrong.
Well, we'll be in touch about that.
Okay.
Let's see. And I'm going to here pull up the website and share the link to our next webinar. So here it is. We have part 2, understanding and using the data. And oh, let's see. Yes, so the second part is concepts and data analytic techniques. So the first part will be by Dr. Carla Ban, and I will also be sharing my presentation at the end, which you don't need to stick around for, but we'll give you an opportunity for questions after Carla's talk, you know, in case you don't want to hear mine twice, which I won't be offended. So we hope you can join us for that. Thanks for attending.